Hi guys, in this video I am going to talk on structure and function of IgG, IgM, IgA, IgE and IgD immunoglobulin. Before start this video, I would request to all of you please share this video and please subscribe our channel, and to get more updates from our channel please press the bell icon and don't forget to like our video. So let's start today's topic, structure or function of immunoglobulin, various type of immunoglobulin. At first I am going to talk on structure of immunoglobulin G or IgG. This is the structure of immunoglobulin G. Immunoglobulin G is a monomer in the predominant immunoglobulin class present in human serum, produced as part of secondary immune response to an antigen, and this class of immunoglobulin constitute approximately 75% of total serum immunoglobulin. Immunoglobulin G is the only class of immunoglobulin that can cross the placenta in human, and it is largely responsible for protection of newborn during the first month of life. Because of its relative abundance and excellent specificity towards antigen, immunoglobulin G is the principal antibody used in immunological research and clinical diagnostic. And in our previous video I discussed the structure of immunoglobulin. So let's know about some properties of immunoglobulin G. The molecular weight of immunoglobulin G is near about 150 kilodalton. Heavy chain type gamma, which molecular weight is near about 53 kilodalton. Serum concentration 10 to 16 mg per ml. Percentage of total hemoglobin 75% near about. Glycosylation by weight 3%. And distribution intra and extravascular. And the function is secondary immunoresponse. So, how immunoglobulin plays in immunoresponse? The role of immunoglobulin in the immunoresponse. Immunoglobulin G is the major immunoglobulin in blood, lymph fluid, cerebrospinal fluid, and peritoneal fluid, and a key player in the humoral immunoresponse. Serum IgG in healthy human patient approximately 15% of total proteins beside albumin, enzymes, other globulins, and many more. The Fc portion of immunoglobulin G, not FAB fragments, can cross the placenta of a mother and enter fetal circulation, proving the fetus with postpartum protection. Immunoglobulin G are able to react with Fc gamma receptors that are present on the surface of macrophages, neutrophils, and natural killer cell, and can activate the complement system. The binding of Fc portion of immunoglobulin to the receptor present on phagocyte is the critical step in the opsonization, and phagocytosis of particles coated with immunoglobulin G antibody is a vital mechanism that cell use to cope with microorganisms. And immunoglobulin G is produced in the delayed response to an infection and can be retained in the body for a long time. The longevity in serum makes immunoglobulin most useful for passive immunization by transfer of this antibody, and detections of immunoglobulin G usually indicates a period infections or vaccination. And there are four subclasses. Four IgG subclasses described in human, mouse, and rat. The subclasses differ in the number of disulfide bonds and the length of the flexibility of the hinge region. Except for their variable regions, all immunoglobulins within one class share about 90% homology, but only 60% among classes. And IgG1 comprise 60 to 65% of total main subclasses of immunoglobulin G and is predominantly responsible for the thymus-mediated immunoresponse against protein and polypeptide antigen. Immunoglobulin G1 binds to the Fc receptor of phagocytic cells and can activate to complement cascade via binding to C1 complex. IgG1 immunoresponse can already be measured in newborns and reach its typical concentration in infancy. And a deficiency in IgG1 isotope is typically a sign of hypogammaglobulinemia. IgG2, second largest of immunoglobulin G isotopes, comprise 20 to 25% of the main subclasses and is the prevalent immunoresponse against carbohydrate or polysaccharide antigen. Adults' concentrations are usually reached by 6 or 7 years old. Among all IgG isotope deficiency, a deficiency in IgG2 is the most common and is associated with recurring airway or respiratory infections in infants. IgG3 comprise around 5 to 10% of total immunoglobulin G and plays a major role in the immunoresponses against protein or polypeptide antigen. And the affinity of IgG3 can be higher than that of IgG1. And IgG4 comprise usually less than 4% of total immunoglobulin G. Immunoglobulin 4 does not bind to the polysaccharide. In the past, testing for immunoglobulin G4 has been associated with food allergies, and recent studies have shown that elevated serum levels of immunoglobulin G4 are found in patients suffering from sclerosing pancreatitis, cholangitis, and interstitial pneumonia caused by infiltrating IgG4 positive plasma cell. The precise role of immunoglobulin G4 is still mostly unknown. Now I am going to talk about the structure of immunoglobulin M or IgM. Serum immunoglobulin M exists as a pentamer in mammals and comprises approximately 10% of normal human serum immunoglobulin content. It predominates in primary immunosuppression or primary immunoresponses to most antigens and is the most efficient complement fixing immunoglobulin. IgM is also expressed on the plasma membrane of B lymphocytes as a monomer. In this form, it is a B cell antigen receptor with the heavy chains, each containing an additional hydrophobic domain for anchoring in the membrane. Monomers of serum immunoglobulin M are bound together by disulfide and joining J chain. Each of the 5 monomers within a pentamer structure is composed of 2 light chains, either kappa or lambda, and 2 heavy chains. Unlike the heavy chains in immunoglobulin M, monomer is composed of 1 variable and 4 constant regions, with the additional constant domain replacing the hinge region. IgM can recognize epitopes on invading microorganisms leading to cell agglutinations, and this antibody antigen immunocomplex is then destroyed by complement fixations or receptor mediated exocytosis by macrophages. And IgM is the first immunoglobulin class to be synthesized by a neonate and plays a role in the pathogenesis of some autoimmune disease. There are some properties of immunoglobulin M. That is, molecular weight of immunoglobulin near about 900 kilodalton. Uh, Heavy chains type mu and the molecular weight of heavy chains near about 65 kilodalton. Serum concentrations in blood 0.5 to 2 mg per ml. Percentage of total immunoglobulin near about 10%. Glycosylations by weight near about 12%. Distribution mostly intravascular. And function of immunoglobulin is primary response, primary immunoresponse. And the role of immunoglobulin M in immune system is described here. Immunoglobulin M is the third most common serum immunoglobulin and takes one or two forms. That is a pentamer where heavy chains are identical and all light chains are identical. And a monomer, example found on B lymphocyte or B receptors. The large pentameric structure allows for binding of beads between encountered epitopes on molecules that are too distant as to be connected by smaller immunoglobulin G antibodies. And IgM is the first antibody built during an immune response. It is responsible for agglutinations and cytolytic reactions since in theory its pentameric structure gives it 10 free antigen binding sites as well as it possesses a high avidity. Due to conformational constants amounts to the 10 FA fractions or fragments, IgM only has valence of 5. Additionally, IgM is not as versatile as IgG, however, it is vital importance in complement activations and agglutinations. And IgM is predominantly found in the lymph fluid and blood. It is very effective neutralizing agent in the early stages of disease. Elevated levels can be sign of recent infections and exposure of antigen. And the side described that IgM is the first antibody built during an immune response and IgG plays a role in secondary response. Now the structure of IgA. IgA exists in serum in both monomeric and dimeric forms. Comprise approximately 15% of the total serum immunoglobulin. Secretory IgA, a dimer, provides the primary defense mechanism against some local infections because of its abundance in mucosal secretions, that is salivary or tears gland, from tears gland. And the principal function of secretory immunoglobulin A may be not to destroy antigen but to prevent passage for foreign substances into circulatory system. And some properties of immunoglobulin A are here. Molecular weight of immunoglobulin A near about 320 kilodalton with secretory immunoglobulin A. H chain type alpha and molecular weight near about 55 kilodalton. Serum concentration 1 to 4 mg per ml. Percentage of total immunoglobulin near about 15%. Glycosylations by weight 10%. Distribution intravascular and mucosal secretions. And the function protect mucous membrane. And the role of immunoglobulin A in the immunoresponse comprises approximately 15% of all immunoglobulin in healthy serum. Two immunoglobulin A subtypes exist in immunoglobulin A1 and immunoglobulin A2, while mice have only one subclass. They differ in the molecular mass of heavy chain and in their concentrations in serum. And immunoglobulin A serum is mainly monomeric. But in secretions such as saliva, tears, colostrum, mucus, sweat, and gastric fluid, immunoglobulin A found as a dimer connected by a joining polypeptide. Most immunoglobulin A present in secreted form and this is believed to be due to its properties in preventing invading pathogen by attacking the penetration epithelial surfaces. And IgA is a very weak complement activating immunoglobulin, hence it does not induce bacterial cell lysis via the complement system. However, secretory immunoglobulin A works together with lysozyme. Uh, Lysozymes are present in many secreted fluids which can hydrolyze carbohydrate in bacterial cells and thereby enable the immune system to clear the infection. And immunoglobulin A is predominantly found on epithelial cells of surfaces where it acts as a neutralizing agent. And there is some immunoglobulin A deficiency in disease such that the most prevalent antibody defect in selective immunoglobulin A deficiency, alternations in IgA1 or IgA2 ratio often go hand in hand with specific disease states such as recurring infections of the airways or a kidney disorder called IgA nephropathy. And there are various several health conditions that can lead to low levels of IgA in the body, one of which is gonorrhea. This bacteria that cause gonorrhea produce an enzyme that splices IgA antibody into Fc and FAB fragments. The FAB parts still can recognize the bacteria, but without the presence of Fc fragment, attachment to phagocytic cell is not possible here. When the body does not have sufficient quantities of IgA, the person may be diagnosed with selective IgA deficiency. And patients who suffer from a selective IgA deficiency are more prone to autoimmune disorder like rheumatoid arthritis, lupus, allergies, and asthma. And there are some classes of immunoglobulin A. That is immunoglobulin A1 comprise approximately 85% of total immunoglobulin A concentration in serum. Although immunoglobulin A1 shows a broad resistance against several proteases, there are some that can affect or place on the hinge region. IgA1 shows a good immune response to protein antigens and to a lesser degree polysaccharides and lipopolysaccharide. And IgA2 representing only up to 15% of total immunoglobulin A in serum plays a crucial role in the mucosa of the airways, eyes and the gastrointestinal tract, which is GI tract, to fight against polysaccharide and lipopolysaccharide antigens. It also shows good resistance to proteolysis and many bacterial proteases, supporting the importance of IgA2 in fighting bacterial infection. And there are also present polymeric immunoglobulin A and secretory immunoglobulin A. Now I am talking about the structure and function of immunoglobulin E or IgE, and this is the main structure where variable region of heavy chain, variable region of light chain, constant region of heavy chain 1, 2, 3 and 4, and constant region of light chains are present. Light chains may be light chains either kappa or lambda, and heavy chains is epsilon. That is the main overall structure of immunoglobulin E. And there are some properties of immunoglobulin E. Molecular weight of immunoglobulin is 200 kilodalton, heavy chains type epsilon, and the molecular weight near about 73 kilodalton. Serum concentration near about 10 to 400 mg per ml. Percentage of total immunoglobulin is approx 0.002%. Glycosylations by weight is near about 12%, and the distributions in our body, basophils and mast cells in saliva and nasal secretions, and the functions is the protective part against parasites. IgE are found in serum in much smaller quantities than other immunoglobulins. IgE primarily defends against parasitic invasions and responsible for allergic reaction. And the role of IgE in the allergic response. The heavy chain of IgE contains an extra domain by this extra domain CH4. We can see in others immunoglobulin there are constant region of heavy chains are present 3, 1, 2 and 3, but here one extra domains are present. So this extra domain, what is the function of the extra domain? By which it attacks with the high affinity to Fc epsilon receptor 1 found primarily on eosinophils, mast cells and basophils. When antigens such as pollen, venoms, fungus, spores, dust mites or pet danger uh, binds with this the FAV portions of the IgE attached to the cell. The cells degranulated and release factor like heparin, histamine, proteolytic enzymes, leukotrienes, and cytokines. As a consequence, vasodilations and increased small vessels permeability causes fluid to escape from capillaries into the tissues and leading to the characteristic symptoms of an allergic reactions. Most of these typical allergic reactions like mucus secretions, sneezing, coughing, or tear productions are considered beneficial to expel remaining allergens from the body. And in this video, the last topic is the structure and function of immunoglobulin D. This is the structure of immunoglobulin D, where two chains are present, that is light chains and heavy chains. In light chains there are two regions are present, that is variable regions and constant region, and in heavy chains there are also two regions are present, that is variable regions and constant regions, heavy chains 1, 2 and 3 respectively, and the light chains either kappa or lambda and heavy chains is delta. And there is some properties of immunoglobulin D. That is, molecular weight is near about 180 kilodalton, heavy chains type delta, and the molecular weight of heavy chains is near about 70 kilodalton. Serum concentrations is 0 to 0.4 mg per ml approx, and percentage of total immunoglobulin is 0.2% near about, and glycolations by weight is 13% approx, and the distribution lymphocyte surface it is present in lymphocyte surface, and the function of immunoglobulin D is still unknown. Immunoglobulin D are found in serum in much smaller quantities than other immunoglobulins. And membrane immunoglobulin D is receptor for antigen found mostly on mature B lymphocytes. And recent study shows that this immunoglobulin D acts with immunoglobulin M. From this video, I am discuss about the 5 primary classes of immunoglobulin are IgG, IgM, IgA, IgD, and IgE. These are distinguished by the types of heavy chains found in the molecules. And IgG molecules have heavy chains known as gamma. IgM molecules have heavy chains known as mu chains, and IgA molecules have heavy chains known as alpha. IgE molecules have heavy chains known as epsilons, and IgD molecules have heavy chains known as delta. Differences in heavy chains polypeptides allow these immunoglobulins to function in different types of immunoresponses and at particular stages of the immunoresponses. The polypeptide protein sequences responsible for these differences are found primarily in Fc fragments, while there are 5 different types of heavy chains are only 2 main types of light chains, that is kappa or lambda. Antibody classes differ in valency as a result of different number of Y-like unit that monomers that join to form a complete protein. For example, in humans, functioning immunoglobulin M antibodies have 5 Y-shaped units, that is pentameric structure containing a total of 10 light chains, 10 heavy chains and 10 antigen binding site. So this video will help you to get more knowledge on the primary classes of immunoglobulin. If this video will help you, please share this knowledge with your friends. And please tell them subscribe our channel and press the bell icon to get more updates from our channels. Thank you. Thank you from Knowledge of Life, and don't forget to like our videos.